Hello students, we have started our topic from Bio 1, cell unit of life, and from that in our previous lecture we have seen the content up to the plastid. In the plastid we have seen that there are three types of the plastids: leucoplast, chromoplast and the chloroplast. So last part of the plastid, that is the chloroplast, we have seen it with the help of diagram, and in that we have seen what is meant by thylakoid, what is meant by granum, or actually where the light and dark reaction occur in this chloroplast, how the outer and inner membranes are there, both membranes are present, that's why it is called as the double membrane structure. Okay, now in today's lecture we will discuss the next part of this topic, and that is the ribosomes. So ribosomes are present both in prokaryotic and the eukaryotic cell, because in the first lecture we have seen that this is the one of the cell organelle which can be found both in animal, plant as well as in the prokaryotic cell. So this ribosome, if we observe it under the electron microscope, you can observe that there is a granular structure present in the cytoplasm of the all prokaryotic and eukaryotic cell, and that granular structure is nothing but this ribosome. So ribosomes are present in the form of the granular structure. It is present in the form of granular structure. It is not only the granular structure, but it is present in the form of the dense particles. So it is in the form of the dense particles. The granules of these ribosomes are present in the form of dense particle. Then what is meant by dense particle? Dense means भरीव म्हणू शकतो आपण, पॅक असे क्लोज पार्टिकल्स आपल्याला राइबोसोमचे पाहायला मिळतात. सो राइबोसोम्स आर ग्रॅन्युलर अँड दोज ग्रॅन्युलर स्ट्रक्चर्स आर इन द फॉर्म ऑफ डेन्स पार्टिकल. दे आर नॉट hollow one, they are packed full of it, so it is called as dense particle. It was first discovered by scientist Palade. So ribosomes are discovered by scientist Palade. So you have to remember the name of the scientist who discovered this ribosome. It is the Palade. Okay. Now you can observe that ribosomes are of two types, or the ribosomes which we call it as a 80S ribosome and the 70S type of ribosomes. So what is meant by 80S and 70S type of ribosome? Generally 80S type of ribosome is present in eukaryotic cell, while the 70S type of ribosome is present in prokaryotic cell. 80S is present in the eukaryotic cell, while the 70S type of ribosome is present in the prokaryotic cell. And see here, this 80S type has two units. One is called as 60S unit and other one is called as 40S unit, while the 70S ribosome has two units again. One is called as 50S and other is called as 30S. Then what is meant by the 50S, 30S, or the 60S and 40S? So here if we see the structure of any ribosome, whether it is prokaryotic or the eukaryotic, you can observe that here the structure of ribosome is there. This is called as the smaller unit of ribosome, and this is called as the larger unit of ribosome. Okay, can you observe this figure? See here, there are two units of the ribosome. One is called as the smaller unit and another one is called as the larger unit. Such a ribosome is present in prokaryotic as well as in the eukaryotic cell. So the smaller unit of the 70S ribosome is the 30S type, and the larger unit of the 70S type is called as the 50S. So this is the 50S subunit and this is the 30S subunit of the 70S type of bacteria. If we consider it as a eukaryotic ribosome. Then here the larger unit of ribosome may be of the 60S type and the smaller unit of ribosome may be of the 40S type. So here in every type of ribosome you can find the two types of unit. One is called as the smaller unit and another one is called as the larger unit. Okay, so here you have to remember that the two types of the ribosomes in the eukaryotic and prokaryotic cell. Eukaryotic type of ribosome is called as ATS type, which has two units. The smaller one is called as 40S and larger one is called as the 60S. While the prokaryotic cell has the 70S type of ribosome, it has again smaller unit that is called as 30S and larger unit that is called as the 50S. Then here what is the meaning of the S? Here, what is the meaning of the S which is written after 60S, 40S, 50S and 30S? This S stands for Swedberg unit. This S stands for Swedberg unit. Swedberg is the name of the scientist who has given the SI unit, that is called as the Swedberg unit. But we are not going to detail in that unit because you should know only the meaning of S. S stands for Swedberg unit. It is also asked for the MCQ question, that is what is the meaning of S in the ribosome. The meaning of S in the ribosome is called as the Swedberg. Okay, and what it indicate, what the Swedberg unit or what this S indicate? This Swedberg unit indicates the sedimentation coefficient. This Swedberg unit indicates what? The sedimentation coefficient. Okay, then what is mean by sedimentation coefficient? Sedimentation coefficient indirectly states the density and the size of that ribosome particle. Sedimentation coefficient indirectly states the density and the size of the ribosome particle. Okay, so here the ribosomes which are present in the 70S type or the ribosome which is of the 80S type, their sedimentation coefficient is indicated by this S unit. It indirectly states the size and density of that particular ribosome. Because what is meant by sedimentation? There are a tube-like structure. In that tube-like structure, if we put this ribosome and if we put the ribosome in a tube-like structure and if we move that tube-like structure at very fast, that technique is called as a centrifugation technique. At that time you can find that the particles or granules of the ribosome deposit sediment at the bottom of that tubule, and where in the bottom of that tubule they can sediment, that is called as the sedimentation coefficient. So 70S and 80S, in that S indicate the Swedberg unit, and Swedberg unit is nothing but the sedimentation coefficient which denotes the density or the size of that particular ribosome during the centrifugation force, during the sedimentation of that particular ribosome. Okay, so here student, you should know that these ribosomes which are of the 70S or 80S type, both these ribosomes are made up of two important components and they are called as the RNA and the protein. They are the RNA and the protein. So chemical nature of this ribosome is that they are made up of ribonucleic acid, that is the RNA and the protein. Okay, and what is the function of this ribosome? Ribosomes is the site of protein synthesis. All the proteins which are required to our body, which are required to our cell, are synthesized in this ribosome. So what is important from the ribosome? From NEET point of view, ribosomes was first discovered by the scientist Palade. Okay, these are having two units. One is called as a larger unit and other one is called as the small unit. So every ribosome has two units, smaller unit and the larger unit. Then in the prokaryotic cell you will find the 70S type of ribosome, and the eukaryotic cell has the 80S type of ribosome. 70S ribosome has two units, smaller is called as the 30S and larger one is called as the 50S, while the eukaryotic ribosome is of 80S type, which has the smaller unit 40S and the larger unit is the 60S. And question is also asked that the S in the ribosome indicates it is the Swedberg unit, which states the sedimentation coefficient of that ribosome, which indicates the sedimentation coefficient of the ribosome, and that sedimentation coefficient is nothing but the density and the size of particular ribosome. Okay, and the function of ribosome is protein synthesis. If the asked question that what is the chemical composition of ribosome, then it is made up of RNA and protein. So this is about the ribosome, which is the important cell organelle. And one more thing that you have to remember about ribosome is that it is do not have any membrane. So ribosome has no membrane. It has no membrane. If you can remember, there is one more cell organelle we have already discussed, that is the centriole. Centriole also do not have any membrane. Like that, the ribosome is also without membrane, not a single or double, but it do not have any membrane. Okay, so this is also important point you have to note down in the ribosome, that is it is without any membrane. So it is very, very important that you have to remember the membranes which is absent in the ribosome. Then 70S and 80S are the types of the ribosomes. Then S stands for the Swedberg units. These are the important parts we will discuss, or we have discussed from this ribosome. Now next to ribosome, the another cell organelle which is present in the eukaryotic cell particularly, and that is called as the cytoskeleton. That is the cytoskeleton. So second point is cytoskeleton. Okay, see here the meaning of this organelle indicates itself the structure of the cytoskeleton. Okay, you can see that cyto means cytoplasm and skeleton means the network. Okay, so in the cytoplasm of the cell there is a network present that is called as the cytoskeleton. Okay, so see here, if this is the cell, okay, this is the cell. In that cell, at everywhere, this is structure which is called as a cytoskeleton. Whether there central there is a nucleus, outside the nucleus there are various type of cell organelle, and this structure is called as the cytoskeleton. Okay. Then here the fiber like structure, tubular filamentous structure is present in the cytoplasm or the throughout the cytoplasm, which is mainly proteinaceous in nature, which is mainly made up of protein. So their nature, the filament which are present in the cytoplasm are made up of the protein. So it is filamentous, proteinous nature has found throughout the cytoplasm of the cell, that is called as the cytoplasm. That is called as the cytoplasm. Then what is the function of this cytoskeleton which is present in the cytoplasm? So this cytoskeleton has the important function, and those are, so now here we are discussing the functions of the cytoskeleton. This cytoskeleton has various functions, and first function is that it gives mechanical support. It gives mechanical support to cell. See here, any skeleton which is present inside the living organism. If you take the example of the skeleton of the human being. The skeleton which is present in our body gives the support to our body. That's why we cannot collapse. Okay, we can stand properly. We can take our movement properly because the skeleton maintains the shape of our body, and like that it gives overall the support to our body. Like that, this cytoskeleton which is present in the cytoplasm gives the mechanical support to that particular cell. That's why that cell cannot be easily broken down, or that cannot be collapsed. Okay, next one, it is responsible for the motility of cell. Motility means movement, whether that cell can move with the help of the cytoskeleton, or the fluid which is present inside that cell can be moved with the help of this cytoskeleton. So motility means movement, any type of movement, whether it is a movement of cell or it is a movement of the internal structure of the cell, it is due to the cytoskeleton. So the second function of the cytoskeleton is the motility. Then the third function of the cytoskeleton is it gives or it maintains the shape of cell. It maintains shape of cell. Cell shape, like the skeleton maintain the shape of the human being. In our face there is skeleton, in our hand, legs, in our whole body except the tongue, in all part of our body skeleton will be there, and that skeleton maintain our proper shape. Like that, this cytoskeleton maintain the shape of that particular cell. So here the cytoskeleton is important because it is responsible for the motility of the cell. It gives mechanical support to the cell, and it maintains the proper shape of the cell. This cytoskeleton is nothing but it is present in the form of filamentous proteinaceous structure which is present throughout the cytoplasm, and that's why it is called as the cytoskeleton. Okay, now the last part of today's lecture is the cilia and the flagella. Is the cilia and the flagella. So cilia and flagella, these structures we have also seen during the prokaryotic cell, but the cilia and flagella that we are discussing here are different than the prokaryotic cell. Here we are discussing the cilia and flagella in the eukaryotic cell. We are discussing here it in eukaryotic cell. Okay, so see here, cilia is the plural and flagella is also plural. So singular is cilium and singular is flagellum. So singular cilium, plural cilia. Singular flagellum, plural flagella. So what is this cilia and flagella, or cilium and flagellum? This cilium and flagellum are the hair-like outgrowth or hair-like structure from the cell membrane. So these both are the outgrowths of cell membrane. Okay, from the plasma membrane or the cell membrane, you can observe in the eukaryotic cell there are some outgrowths, out of which some are called as flagella and some are called as the cilia. Now if we see the structure of particular cilia, okay, so if we see the cilia, these cilia are the hair-like outgrowths. These cilia are they are hair-like outgrowths, okay, and they work like the oars. They work like oars. For what? either cell or fluid around the cell. Okay, now the cilia are the outgrowths of this cell membrane. So see here, this is structure of the cell. This is the cell membrane, and from this cell membrane there are hair-like outgrowths. These outgrowths we call it as a cilia, and these cilia work like the oars. Oars means what? पाण्यामध्ये आपण जेव्हा बोटमधून जात असतो तेव्हा त्या ठिकाणी एक शाफ्ट असतो, वूड लाईक, वूडचं शाफ्ट, नॉट वूड लाईक, वुडन शाफ्ट इज देअर अँड इट इज यूज फॉर प्रोपेलिंग ऑफ दी बोट. म्हणजे पाण्यामध्ये ते लाकडी ओंक घातल्याशिवाय ती बोट पुढे सरकत नाही, आणि त्याला म्हणलं तर ओर्स. Like that, cilia works, and so here called as a cilia works like the oars, which is particularly for the movement of the cell or the fluid present around the cell. त्यामुळे फ्लुइड जे सेलच्या मध्ये किंवा सेलच्या बाहेर आहे त्याची मूवमेंट होण्यासाठी किंवा सेल प्रॉपर मूव्ह होण्यासाठी या सिलियाची मदत होते. सो लोकोमोशन इज द इम्पॉर्टंट फंक्शन ऑफ द cilia in a cell because it works like the oar. It is responsible for movement of the cell, or it is responsible for the movement of the fluid around the cell or inside the cell. So this is about the cilia, which is the one of the outgrowth of the cell membrane. Now the second one is flagella. Flagella are comparatively longer. Flagella are comparatively longer, okay, and they are also responsible for cell movement, because like the cilia, flagella are also involved for movement of the cell. These are comparatively larger than the cilia, but their function is the same like that of the cilia, that is, it is also responsible for the movement. We have seen the structure of flagella in the prokaryotic cell, but see here the structure of the flagella in prokaryotic cell is different than the structure of flagella in the eukaryotic. So here we are discussing the flagella of the eukaryotic cell. It do not have the same structure like that of the prokaryotic cell. So here we have discussed the flagella part of the cell, and what we have to remember from the cilia and flagella, both the cilia and flagella are responsible for the movement of the cell. This point is very, very important. If the question is asked that oar-like structure is performed by, the answer is of the cilia. And now student, see here, there is the one diagram is you can see on the board, and under that the label is there, that is a section of cilia and flagella. Section of cilia and flagella. If under the electron microscope this cilia or this flagella is observed, then you can observe the structure of that cilia and flagella same. And what you can observe here, see here, this is the section of the cilia and flagella. You have to study diagram very carefully, because from this diagram number of questions are asked for the NEET point of view. And see here what is given here. So under the section or under the electron microscope, if we observe the cilia and flagella, you can see that the outer membrane of the cilia and flagella is called as the plasma membrane. So in the section, the cilia and flagella shows outer membrane, and that outer membrane is called as the plasma membrane. Okay. The core means central part. Core means central part. So core of the cilia and flagella is called as the axoneme. Is called as the axoneme. Central part, nothing but the core of cilia and flagella is called as axoneme, and in this axoneme you can observe the 9+2 arrangement. You can observe the 9+2 2 arrangement. Then what is mean by 9 plus 2 arrangement? So here you can observe that in the core of cilia and flagella, or in the axoneme of cilia and flagella, you can observe there are doublets of 9 peripheral microtubules which are radially arranged. So here doublets, these are the round part that we are observing here. These are the in the form of doublet. These are the microtubules which are radially arranged. Okay, so these are 9 microtubules. What is mean by microtubules? Micro means very microscopic tubular filamentous structure which is present in the cell, that is called as microtubules. So inside the cilia and flagella, you can observe at the periphery there are doublets of 9 microtubules which are radially arranged. Radial means in a circular form. If we count this microtubule, see here 1, 2, 3, 4, 5, 6, 7, 8 and 9. Okay, so there are the total 9 microtubules which are present in the form of this doublet, and they are peripherally arranged in the radial form in the section of the cilia and flagella. Okay, while at center there are 2 microtubules which are also in a pair. So here it is given as a 2. So 9 in the form of doublet, and centrally there are 2 microtubules. So 9 plus 2 arrangement is there in the section of the cilia and the flagella. Okay, now what we have to remember again, these two central microtubules. So here in the figure name is given that, here name is given that is the central microtubule. Name is given central microtubule. These are the 2 central microtubules which get connected to each other by a bridge. This is the central bridge which connect these 2 central microtubule. Okay, and this central 2 microtubule is also has a covering, and the name of that covering which is shown by yellow chalk, and it is called as the central sheath. So central sheath is the covering which cover the central 2 microtubules. Okay, now then the peripheral microtubules which are present in the doublet form, these peripheral microtubules get attached to this central sheath by the radial spoke. See here what I am saying, the peripheral doublet microtubule get arranged or get attached, not arranged, get attached to the central sheath by a radial spoke. Radial spoke ना हे peripheral doublet microtubules तुम्हाला कुणाला अटॅच झालेले पाहायला मिळतात, central sheath ला. Okay, now here the names are plasma membrane, peripheral microtubules which are in the presence of doublet, central microtubules, radial spoke, central sheath, and the one name is given that interdoublet bridge. Here name is given that interdoublet bridge. Interdoublet bridge means 2 nearby doublet of microtubules get connected to each other by this interdoublet bridge. So interdoublet bridge connect 2 successive doublet microtubule. जवळजवळचे 2 डबलेट मायक्रोट्यूब्यूल कनेक्ट करण्याचं काम कोण करतंय, इंटरडबलेट ब्रिज. सो अकॉर्डिंग टू दिस फिगर which points are important here, there is question is that in the section of the cilia and flagella what is the arrangement of the microtubule, then you have to remember that there is the arrangement of 9 plus 2. Okay, second question, what is the core of cilia and flagella? Core means the central part of cilia and flagella. The core of cilia and flagella is called as the axoneme. So student, in the circular form, the structure that we are seeing, we are watching here, that is called as axoneme. So all the structure which is present inside the cell membrane is called as the axoneme. So axoneme is the central part or the core part of the cilia and flagella. So this 9 plus 2 arrangement of microtubule you can observe inside this axoneme. You can observe inside this axoneme. Then next part is that how the microtubules are arranged in the axoneme. So there are 9 microtubules which are present in the form of doublets. There are 9 microtubules which are present in the form of doublets. They are arranged at the periphery of the axoneme. They are arranged in the radial form in the axoneme, okay, while the 2 microtubules are present at the center. 2 microtubules are present at the center. So it is 9+2 arrangement. Next point is that central doublets are connected to each other by bridge, as well as central doublets are covered by central sheath. Okay, central doublets central 2 microtubules are covered by central sheath, and central 2 microtubules are connected to each other by one of the bridge. Okay, while the peripheral 9 doublets are connected to the central sheath by radial spoke. Peripheral 9 doublets are connected to the central sheath by the radial spoke, while the peripheral doublets are connected to each other by the interdoublet bridge. They are connected to each other by the interdoublet bridge. Student, the important thing is that you have to first understand the diagram of the section of cilia and flagella. If the cilia and flagella we can observe under the electron microscope, then both this cilia and flagella shows such a type of structure. For the NEET exam, there may be diagram given, and according to which you have to label the particular diagram, or the question is asked that what is the outer covering of cilia and flagella, then you have to give the answer plasma membrane. The successive doublet bridge at the periphery are connected to each other by, then you have to write the interdoublet bridge. If the question is asked that what is the name of the central microtubule, then you have to which covering is present around the central microtubule, then which you have to answer the central sheath. So student, see here, this is very important part of the cilia and flagella, and by this diagram you can understand very well the concept of cilia and flagella. So in today's lecture we have completed 3 small cell organelle. One is called as ribosome. Important thing is that it is without any membrane bound structure. Second thing is that the cytoskeleton, you have to remember the functions of the cytoskeleton, that is, it gives mechanical support to cell, it is responsible for motility of the cell, and it maintains the shape of the cell. And third cell organelle, that is cilia and flagella, whose structure is totally different from the bacterial cell. This cilia and flagella, if cut under the if cut and observed under the electron microscope, then you can observe the structure of this cilia and flagella. Do not practice the cilia and flagella for sketching, because you have to not draw this figure, but you have to remember the labeling of this figure and the concept of this figure. So students, here we have completed these 3 important cell organelle. The next part of the last part of this cell unit of life will be discussed in the next lecture. Thank you very much.